Hi, seventh graders. This is Mrs. Hudgens. I'm going to talk to you a little bit about some of the objectives that we've been studying recently, just to make sure you're all understanding. So last week we were wrapping up genetics, and this week we're going to be moving into biotechnology. So, for the past couple of weeks, we've been looking at this objective. So, you should know how to develop and use a model to describe why asexual reproduction results in offspring with identical genetic information, and sexual reproduction results in offspring with genetic variation. So, really, to sum all of that up, if you look in the left corner, we talked about the process of mitosis. So, organisms that undergo asexual reproduction perform a process very similar to mitosis, in which they are cloning themselves. So, they are coming up with they start with the right number of chromosomes and they end up with the exact duplicates of the same amount. So it's the same type of genetic information that they're passing to their offspring, and there's only one parent. If you look next to that, meiosis occurs in the sex cells. So it splits the chromosome number in half. So half of the chromosomes. So if we're talking about humans have 46 chromosomes or 23 pairs. So of chromosomes. So in meiosis, the egg would then be split down to 23 chromosomes and the sperm would be 23. So when fertilization takes place and the sperm and egg unite, that's going to give 46 chromosomes. So each parent has a say in what the offspring look like, their genetics. They're passing that on. So if you look on the right, we looked at how by knowing the genotypes of the parents, we can find the probability of what the offspring's traits and what their characteristics could be. Okay. And so, again, to push that objective home, genetic variation is going to come from sexual reproduction because we have two parents that are passing on their genetic information. And with asexual reproduction, it's only one parent. So it's a clone and there is no genetic variation. Last week, in addition to finishing up on Punnett squares, we looked at mutations and how mutations can occur in DNA. So our objective was to develop and use a model to describe why structural changes to genes, mutations, located on chromosomes may affect proteins and may result in harmful, beneficial, or neutral effects to the structure and function of the organism. So you really dove into the structure of DNA and how it's composed and the importance of the base pairs and what kind of protein they will code for to help an organism live. And so, since DNA replicates itself over and over to make new cells, we have mutations that occur. So, a lot of times, those mutations may not have any effect on the protein that's made. If there is a negative effect on the protein, we would say that was harmful to the organism. And if it is helpful to the organism, then we would say it's beneficial. So I want to show you a quick clip about a mutation story, which kind of is beneficial in some parts and then harmful in others. So, and that's on PBS Learning Media.
For thousands of years, coastal West Africans have fished the sea to make a living. But 4,000 years ago, a deadly disease arrived here, bringing death and misery. Its name, malaria. That people continue to live and fish here today demonstrates the power of evolution. power felt in the widespread effect of just one mutation in a single gene. Malaria is caused by a tiny parasite carried by mosquitoes. Humans become infected with the parasite through a mosquito's bite. It may take just a few short weeks for the parasite to destroy a human's red blood cells, necessary for spreading oxygen throughout the body. A red blood cell is invaded by the parasite, which soon multiplies, causing the cell to burst. These new parasites are now free to invade yet more cells. In West Africa, malaria is so common that most children are infected with the disease. Though many suffer symptoms severe enough to warrant trips to the hospital, like this one in Senegal, for most, the disease is not fatal. The key to their resistance is in their genes. Dr. Ronald Nagel studies the genes that make hemoglobin an essential part of red blood cells. Genes are all paired, with each parent supplying one half of each pair. If either hemoglobin gene undergoes a mutation, the hemoglobin it makes will be changed. This particular mutation, called the sickle cell gene, is tiny, but it's enough to change the shape of the hemoglobin molecules it makes.
People that had one copy of the gene were able to survive their infection, the malaria infection. They would not die of it. Hence, they were able to grow up, get married, have children, and pass the genes on to the next generation. This is selective pressure. That gene had an advantage in that particular environment for those carrying it.
We all have lots of small gene mutations. Mostly they go unnoticed. But if the environment changes, one of them may suddenly show unforeseen effects, good or bad. In this case, one copy of the gene is beneficial, but two can be disastrous, as Fatu and Sheikh Assar found when they started a family. Both Fatu and Sheikh are protected from malaria because they each have a single copy of the sickle gene. Their children each had a 50% chance of receiving one single copy of the mutated gene. Or 1 in 4 might get both parents' normal genes, leaving that child unprotected. Another 1 in 4 might inherit both sickle genes. And that is the case for their daughter Fatu, named for her mother. Because she inherited both copies of the sickle cell gene, all of Fatu's hemoglobin is adversely affected, not just half. Her molecules turn into rigid rods that provoke sickling of the red blood cells, even in the absence of malarial parasites. The slightest exertion can cause a sickling emergency. Her cells sickle one after another, causing blockages in her narrow blood vessels. Fatu's blood flow is cut off, and she experiences agonizing pain. The sickling caused by the mutation led scientists to name this disease sickle cell anemia. Fatu's life will be far from comfortable, but medicine can help her survive this debilitating disease. Though the sickle defect was a mistake, causing great harm to some, for others it is priceless protection against malaria. This is how evolution works. Mutations occur at random and may or may not have positive consequences.
The more diverse people are, the more likely that population is going to survive, even to things that might come in the future, infections that we don't know about now. Some people might be by chance more resistant to that future infection that might wipe out a population. If everybody's the same, if everybody has the same genetic background, a population can be easily wiped out by a single plague.
Okay, so that showed you. So, if they were heterozygous for the trait of sickle cell anemia, it was helpful. Um, it allowed those people to live and survive and reproduce. But in the little girl's case, she had both. And so that was a genetic disorder that is very harmful to her. So that mutation, having both of those genes for that recessive trait, has been harmful to her. So now this week, we're moving on, and our new objective this week for biotechnology is gather and synthesize information about the technologies that have changed the way humans influence the inheritance of desired traits in organisms. So, you will be looking into biotechnology. So, this is advancements in genetics, um, taking the knowledge that we have about genetics, about the human genome, and modifying food, modifying organisms, inserting genes into organisms, being able to clone larger animals. And so, this I think you will find very interesting. And so, have a good time investigating this. I have extra materials for you to look into on my website. And if you have questions, let me know. Have a great week.